1. The journey begins. Fertilization. The process starts with the fusion of gametes to form a single-celled, totipotent zygote. Pre-implantation period. This stage covers the development within the oviduct as the embryo moves toward the uterus. Morphological transition. The embryo transforms from a solid ball of cells into a specialized fluid-filled structure. 2. Initial cleavage divisions. Rapid cell cycles. The zygote undergoes successive mitotic divisions called cleavage, increasing cell number without increasing total volume. Blastomere formation. The individual cells produced during these early divisions are known as blastomeres. Maternal control. Initial divisions are largely driven by mRNAs and proteins stored within the oocyte prior to fertilization. 3. Cell numbers in mammals. Human counts. By the blastocyst stage, a human embryo typically contains between 100 and 150 total cells. Mouse counts. The laboratory mouse reaches the blastocyst stage with approximately 64 to 100 cells. Large mammals. Bovine and porcine embryos often reach higher counts, ranging from 120 to over 200 cells at the time of cavitation. 4. Symmetry and identity. Symmetric divisions. The first few divisions are generally symmetric, producing daughter cells that are morphologically and potentially identical. Totipotency. At the 2-cell and 4-cell stages, individual blastomeres often retain the ability to form an entire organism if isolated. Stochastic variation. While appearing identical, subtle differences in protein distribution begin to emerge as early as the 4-cell stage. 5. Breaking symmetry. Asymmetric division. As the embryo reaches the 8 to 16 cell stage, divisions can become asymmetric, creating one polar and one nonpolar daughter cell. Apical basal polarity. The inheritance of the apical cap determines whether a cell stays on the outside or is pushed to the interior. Positional information. Cells moved to the interior are destined to become the inner cell mass, while exterior cells become trophectoderm. 6. The compaction process. Maximizing contact. At the 8-cell stage, blastomeres flatten against each other, increasing intercellular adhesion. E-cadherin role. The protein E-cadherin is essential for zipping the cells together to form a solid ball called the morula. Cellular sealing. Tight junctions form between outer cells, creating a permeability barrier necessary for later fluid accumulation. 7. Maternal to zygotic transition. Zygotic genome activation. The embryo eventually shifts from relying on maternal factors to synthesizing its own proteins from its own DNA. Species timing. The timing of this activation varies. It occurs at the 2-cell stage in mice and the 4 to 8-cell stage in humans. Transcriptional overhaul. This transition is a critical genetic milestone where the embryo assumes control over its developmental program. 8. Epigenetic remodeling. DNA demethylation. The embryo undergoes a global wave of DNA demethylation to erase parental imprints and establish pluripotency. Histone modifications. Chromatin structure is rearranged to allow access to genes required for early lineage specification. X chromosome inactivation. In female embryos, the process of balancing gene dosage between sexes begins during these early stages. 9. Genetic regulation of the ICM. OCT4 expression. The transcription factor OCT4 becomes restricted to the internal cells, serving as a master regulator of pluripotency. SOX2 and Nanog. These proteins work alongside OCT4 to prevent the inner cells from differentiating prematurely. Antagonistic signaling. The internal genetic program actively represses genes like CDX2, which are required for placental development. 10. Formation of the blastocyst. Caviton and pumping outer cells use sodium pumps to move fluid into the center of the morula, creating a cavity called the blastocoil. Expansion. The accumulation of fluid creates internal pressure, causing the embryo to expand and thin the zona pellucida. Eccentric positioning. The inner cell mass is pushed to one pole of the fluid-filled sphere, defining the embryonic-abembryonic axis. 11. Maternal versus embryonic proteins. Initial reliance proteins for DNA replication and early metabolism are almost exclusively maternal in origin. New protein synthesis. Following genome activation, the embryo produces its own structural proteins and signaling molecules. The shift. By the blastocyst stage, the majority of the proteome is derived from the embryo's own genetic instructions. 12. Summary of ICM development. Lineage segregation. The ICM represents the first successful separation of the embryo proper from extra-embryonic tissues. pluripotency reservoir. These cells are the source of embryonic stem cells capable of forming all three germ layers. Critical milestone. The successful establishment of the ICM is a prerequisite for implantation and further post-implantation survival.